Hello, my lovely biology humans. Okay, topic 2.6. Will topic 2 ever end? The answer is no. That's a long topic, right? Lots of molecules to cover here. Okay, so at the beginning of topic 2, we started talking about those 4 macromolecules for biomolecules that are essential for all life, right? You've got carbohydrates, you've got lipids, we've got proteins we just spent a bunch of time on. And the very last group is nucleic acids, right? So looking at deoxyribonucleic acid and ribonucleic acid. So our DNA and our RNA. So lots of, again, molecular structures that we need to know for nucleic acid, more drawing, and knowing the parts of it and then the differences between the two in our body. So we'll break this up into 2 parts. We'll focus on kind of like the molecular structure in the first part and then the differences between the two in the next. Okay, so you probably have I know you have some exposure to DNA and RNA from bio, and plus just like how we talk about it all the time. But now we're going to talk about so we've seen these little twisted helixes. We've seen the RNA. You've learned double strand and single strand. But now we're going to look at the actual differences on the molecular level, because you've learned all these things so far, right? So we've learned about nitrogen bases. We've learned about hydrogen bonds. We've learned about molecule rings. So it makes more sense to talk about this last after you have all this stuff kind of in your head, even though our DNA is that structure to make all those proteins that we just learned about. Okay, we'll also talk about some scientists, their research, and how they came about with the structures of DNA and RNA and all that jazz. All right, so let's go into the basic of a nucleotide, right? So a nucleotide, we're going to remember that nucleotide is the building blocks of my DNA and my RNA. Okay, we've already looked at this word monomer, so make sure you have that. Remember that the monomer is the single unit, and then the polymer is when they're put together. So my nucleotide is my single unit. My polymer is DNA and RNA. Okay, so my monomer of a nucleotide looks like this. All right, so we can kind of draw it up here, and I'm pretty sure your book does this too, right? So you've got your bases, you've got your sugar, and you've got your phosphate group. Okay, so this is the basic structure. We'll learn how to draw these in class, right? So of course, there's instructions on how to draw things in class, but this is my nitrogen base. My nitrogen base can be either A, T, G, or C and U. We'll learn about those base pairing rules. So these are my adenine, cytosine, guanine, thymine, uracil, right? These are my nitrogen bases. You will need to know all those. These are the ones that pair up in the middle, right? That create those base pairing, A, T, G, C. In the middle here, I have this 5 carbon sugar carbohydrate, and this is my 5 carbon. You do need to know the 3 and the 5. We'll talk about those a little bit later, but I do have my 3 carbon and my 5 carbon here. So see right here, and this is kind of the 2 that links. This is the sugar that links the nitrogen base to the phosphate group. So way out here, I've got my phosphate group. So here's my phosphate. These phosphates are responsible for the bonds on the outside, so you do need to know those portions. But for now, just know base, right? That pentose, pent meaning 5, and then this phosphate group out here. All right, so those are my basics of the nucleotide. Okay. Now, DNA and RNA. So looking at those 2 things, go ahead and write this chart into your notes. Right, and so this pentose sugar, go ahead and write this down for now. So that 5 rings in there, deoxyribo and ribose. So you do need to know that this deoxy right here, right? It's not di meaning 2, it's D meaning without. It doesn't have that extra oxygen group. Ribose does have an oxygen group. So I've got deoxyribose, that is my type of sugar, right? So anytime we see that ose, glucose, right? Fructose, though we're going to think sugars, carbohydrates, all right? The nitrogenous bases, so the different nitrogen bases, right? We learned about the 4, A, T, G, and C. However, you do need to know, and then RNA, so when I make that RNA, T cannot leave the nucleus, so it actually is replaced with the base uracil. Uracil is not in my DNA. For strands, we need to know that DNA is double. It's got the double-stranded helix. My RNA is single. And the basic function. So my DNA has the genetic instructions, and these are the master. It has the full set, right? So all of my DNA is in every single nucleus. My RNA and we'll learn about different types, is a temporary structure that just goes in and copies just a little bit, right? So the RNA is the one that goes like, oh, we need more of this. So I'm going to go into the DNA, right? Just copy this little directions, and then the RNA can actually leave the nucleus, right? So the DNA is bound into the nucleus, the RNA can leave. We'll learn about messenger RNA, transfer RNA, all that fun stuff. But just know the difference, the key structural difference is that lack of oxygen, the double strand versus the single, the different bases, and what their functions are. Okay. All right. So looking at those on the molecule or the molecular level, like I just said, where that oxygen exists, right? So when I'm drawing out, I'll show you in your book where you learn how to draw it out. So I still have this phosphate group here, right? My bases, again, if you kind of this, right? Or here, my bases, I've got T on this side and G on this side. Here's my nucleotide for my ribose. Can we spot the extra oxygen? I can, right? So here's my alcohol here. I'm sorry, that OH group here. And then where their oxygen is, this is the one without it. Okay, so the ribose sugar has that oxygen group, the deoxy does not. Other than that, right, if you cover it up, that phosphate group is the same. The nitrogen bases are the same, except the uracil and the thymine. Okay, in your book, let me show you real quick here, right? So there's this whole section on drawing RNA and DNA, right? So talking about circles and pentagons. So we will practice that in class. Don't worry about drawing those right now. We'll practice all those fun rules that we need to know when in class. Okay. All right, moving on. Let's keep going. So how that forms, right? So we'll practice that drawing, but let's just talk about like what this actually look like. Okay. So what I need to know in these, right, this type of structure here. So, right, I've got my phosphates here and my phosphates here, right? These are the ones that are responsible for this bond that keeps my ribose sugars on the outside of the ladder together, right? So this is the outside of the DNA, right? This is the inside that's going to pair with the other one. So this is the outside, right? These are called phosphodiester bonds, right? So these ones right here are the phosphodiester bonds. They're the bonds that keep those nucleotides on the outside right together. Remember, I told you to pay attention to that 5 and 3, right? So the phosphate group on one attaches to the 3 on the other, that hydroxyl group, right? So I've got the 5 and the 3 coming together, right? I'm sorry, the 3 and the 5 here. So learning the numbers that go around 1, 2, 3, 4, 5, those are the 2 that come together. And when we're talking about copying, you do need to know what the 5 direction and the 3 direction are, right? So we'll look at that in class where those 5 and 3 are. That's just how we number the carbons, so we can pay attention to what comes together. This nucleotide strand will run in the 5' to 3', and those bonds are the ones that keep it together. So that's how the outside comes together. Ooh, I skipped this first one. So knowing that those nucleic acids come together between that bond, it is a condensation reaction. So when I get DNA molecules, those deoxyribose, right, those bases coming together with my my phosphates and my sugars and my bases, they attach via condensation. So water is produced. Okay, we should be kind of familiar there with our condensation and our hydrolysis reactions. Okay, here are my nitrogenous bases, right? So they all have a nitrogen in them. You can kind of pay attention here to where I see all these nitrogens. Do you need to know how to draw these out? No, with all these bonds around, but you do need to know the single and the double rings, okay? So you do need to know right here that my guanine and my adenine are my double ringed purines, right? So I've got 2 rings, carbon rings here, all right? Now my cytosine and my thymine or my uracil are single ringed pyrimidines, right? So these are single rings here. This is just how they're classified. So C, T, and U all have that single ring of the still having my nitrogen bases, right? So lots of nitrogen in these rings, but my A and my G are my double. Okay, both are composed of 4 nitrogen bases, right? You can see how they are 1, 2, 3, and 4 here, versus here they're 1, 2, 3. Where's the 4th one? 1, 2, 3, 4. Oh, right there, right? So I've got that onto the side. And so they have those nitrogen, oh, I'm sorry, hold on. Oh, I'm sorry. I read that wrong. I'm not even going to rewind it. Just, are you listening anyways? The DNA and the RNA are composed of one of four nitrogen bases. There's three in each one. This one's like, where's this one? So 1, 2, 3, 4 on this one, 1, 2, 3, 4 on this one, right? And then 1, 2, 3 on these ones, right? So the different amounts of nitrogen in each. I'm sorry, I didn't mean to confuse you there. These ones have, but there are four different nitrogenous bases: A, T, G, and C. Some are double ringed, some are single ringed. Okay. All right. Let's keep going on here. All right. Let's do two more for DNA and then we'll go to RNA. Okay. So the base pairing rules, right? So I already used that like outside of the ladder analogy, but you can think about this DNA here as like a ladder where I have the outside, they're called the rungs, right? So I've got my phosphates and my sugars here. And then these are like the ladder, right? So the nitrogen. So this analogy is used a lot, right? So just so you can know, like these are the outside of the ladder and these are the inside, right? So it says DNA molecules form double-stranded ladders via these bonds between my bases. My backbone has the sugar and phosphate, and the ladder, the rungs part, are those different bases, all right? You do need to know the complementary base pairing rules, right? So my G and my C pair together, and my A and my T. In between, you see these right here? We've already learned about these. My bases right here are put together with hydrogen bonds. So the hydrogens in those last ones that we looked at, they come together. Remember that hydrogen bonds are weaker, right? So they're those bonds that come together, but they're not really strong. These ones on the outside are made by covalent bonds. So my outsides are a lot stronger than the insides. There's a good reason for that because it has to come apart so that it can be copied and come back together. Okay, my G and my C have 3 hydrogen bonds, right? So you can see those lines I kind of drew through. My A and my T have 2. So G and C have 3, the A and the T have 2. All right, just to kind of put that into perspective, I'm gonna go like this, right? If I come back here to what it actually looks like, my G and my C are put together, one right here and right here, my G and my C, these are my base pairs. With those hydrogens, they've got 3. My A and my T come together with just the 2, right? So you can kind of look at like the biochemistry of how that works, but you need to know the base pairing rules. The hydrogen bonds are weak and that they go together with those 3 and the 2. Okay. All right. Last one for part 1. What you need to know about that ladder, right, is that in order for it all to fit inside the nucleus, right, is that these nitrogen bases, as they go anti-parallel at 5 to 3, 3 to 5, we'll talk about, they start to twist, right? So they start to twist together because they're in those opposite directions and they're attracted to each other. And this is the most, what it's called, right, is the most stable energy configuration. So coming together in that spiral type shape is how the DNA protects itself, and then it has the most stable molecule. So it's not going to be broken apart or react very like easily with anything. Okay, so in that double, in that double helix arrangement, there's approximately 10 to 5 per rotation. So if you look at, oh gosh, like what would it be? This is a rotation, right? We get 10 to 5 per rotation. That's how many are like scrunched into that small one. All right. So that's my structure of my DNA. I've got double, right? Deoxy, double. In my next lecture, part 2, we're going to look at RNA, how it's put together, and then the differences between the two. I will see you later.